Good morning to one and all present here. My topic for presentation is response of bud induction in pineapple over applied plant bioregulators. First of all, we can see about pineapple. Pineapple is an important crop belonging to the family Bromeliaceae, which is a low nutrient requiring crop. This pineapple can be propagated by various methods like seed propagation, vegetative propagation. In seed propagation, the seeds lack dormancy because of the very tough seed coat which is present outside of the seeds. And in vegetative propagation, there are two types, that is conventional methods and non-conventional methods. The conventional method of propagation include using slips, crowns, side suckers and ratoons. But the non-conventional method like stem sectioning, which produce more number of planters from a single stem section, is used in this study. The objectives of this studies is to access the response of stem sections for the production of pineapple propagules in the nursery, to analyze the impact of various growth promoters in activation of buds for the development of plantlets in pineapple, to study the feasibility in rapid multiplication of plantlets with the stem propagation of pineapple. The methodology, that is two factors were involved in this study. First factor is type of stem cuttings and second factor is growth promoters. Type of cuttings include whole stem cuttings, half stem cuttings, sliced cuttings and longitudinal stem cutting. And the growth promoters include IBA, coconut water, tetracycline, chitosan and no treatment. Together it includes 20 treatments with 3 replications, and the design of experiment is FCRD, that is factorial completely randomized design. And the methodology include, first we have to select the plantlets, that is 10 days before the first harvest we have to tag the plants, and after harvest we have to uproot the plants, we have to trim the leaves and roots and completely remove the leaves and expose the buds present in the stem sections. And the stem section has to be treated with the different growth regulators, and it is planted under polyhouse condition. After some days the buds started emerging and it produced plantlets. These plantlets can be used for propagation of the pineapple. Now we can see the experimental results. The first parameter is duration of first axillary bud emergence. That is, the early emergence of buds were seen in S1G2, that is coconut water treated with the whole stem cuttings, that it produced its first bud emergence in 8 days. The duration of axillary bud sprouting, that is sprouting means the leaves production. The first leaves, true leaves were produced early from S1G2, that is within 21 days or 20 days. The whole stem cutting treated with the coconut water has produced its axillary bud sprouts. The next one is number of axillary buds emerged in 15 days after planting. The more number of axillary buds has been produced by S1G2, that is whole stem cuttings treated with the coconut water. Next one is number of buds sprouted 90 days after planting. That is more number of buds, that is 23 buds, nearly 23 buds were produced from whole stem cuttings treated with the tetracycline. And total number of propagules on 90 days after planting. This whole stem cuttings treated with the coconut water has produced nearly 31 buds from a single stem sections. The more number of stem sections produced can be used for the vegetative propagation. And the results and conclusion. The better performance of bud emergence, plantlet production, morphological characters and growth analyses were shown by whole stem cuttings followed by longitudinal stem cuttings. And in case of growth promoters, coconut water has shown positivity for early bud emergence as well as sprouting, dormancy, breaking of more axillary buds. And with regard to interactions, the whole stem cuttings with the coconut water has performed better with the bud growth and plantlet production. And this is the view of the plantlets produced from the stem sections. We can detach the each plantlets and we can produce for the multiplication of these pineapples. Thank you.